Did you know that some of the first antidepressants were discovered by accident while treating tuberculosis? Let's go back to the 1950s. Doctors were treating tuberculosis patients with a drug called iproniazid and noticed something surprising: these patients started to feel happier. It was unexpected and caught everyone's attention.
Then, another surprise came along with an allergy drug called imipramine. Patients reported feeling better emotionally, even though it was meant for allergies. These unexpected mood improvements led scientists to dig deeper.
Both iproniazid and imipramine increased levels of certain chemicals in the brain known as monoamines. Monoamines include neurotransmitters like serotonin, norepinephrine, and dopamine, which are key players in regulating mood, arousal, and emotion.
This led scientists to a revolutionary idea: what if changes in these brain chemicals caused mental health conditions like depression? This is how the chemical imbalance hypothesis was born. It suggested that by adjusting these chemicals, we could treat depression.
Today, the antidepressants we use are based on this hypothesis. They primarily target serotonin and norepinephrine, two important monoamines. Now, let's get into the specifics. We have different types of antidepressants like SSRIs, SNRIs, TCAs, and MAOIs. Each works in its unique way to boost these brain chemicals and help improve mood.
Are you ready to dive deeper into how each of these medications works? First, let's talk about SSRIs, the most commonly prescribed antidepressants. SSRIs, or selective serotonin reuptake inhibitors, work by increasing the levels of serotonin in the brain. They do this by blocking the reuptake of serotonin into nerve cells. But what exactly is reuptake? It's important to understand this concept when studying how neurotransmitters function in the brain. So, let's dive a bit deeper.
Imagine serotonin as a chemical messenger. When a nerve cell releases serotonin, it sends it out into the space surrounding it. This serotonin then travels across that space and attaches to receptors on the surface of nearby nerve cells, where it exerts its effects, like improving mood. However, not all of the released serotonin is used. The unused serotonin needs to be managed, so it attaches to receptors on the surface of the nerve cell that originally produced it. From there, it gets taken back up by the nerve, recycled, and can be released again. This recycling process is called reuptake. It's a way for the brain to maintain a balance of serotonin. Antidepressants like SSRIs work by inhibiting this reuptake process. By blocking reuptake, more serotonin remains available in the synaptic cleft, the space between nerve cells. This increase in serotonin levels helps to improve mood and alleviate depressive symptoms. SSRIs are currently understood to work by attaching to two spots on the transporter, the central site and the allosteric site. This blocks serotonin from binding to the transporter and being taken back into the neuron. Let's do a quick quiz. Do you know how SSRIs work? Correct! SSRIs inhibit the reuptake of serotonin into nerve cells, which increases the level of serotonin in the brain.
Next, let's talk about SNRIs, or serotonin and norepinephrine reuptake inhibitors. SNRIs work by stopping the reuptake process of both serotonin and norepinephrine. These drugs are similar to SSRIs in that they boost serotonin levels in the brain. But SNRIs go a step further by also increasing norepinephrine levels. This dual action not only helps improve mood but also reduces anxiety and enhances focus. One well-known SNRI is duloxetine. What's interesting about duloxetine is that it does more than just affect mood. It also interacts with pain pathways in the spinal cord, making it effective for treating chronic back pain and diabetic nerve pain. Let's do a quick quiz. Do you know how SNRIs work? Correct. SNRIs work by inhibiting the reuptake of both serotonin and norepinephrine. This helps improve mood, reduce anxiety, and enhance focus.
Tricyclic antidepressants, or TCAs, were the first group of medications developed in the 1950s to treat major depression, bipolar depression, anxiety, obsessive-compulsive disorder, and other psychiatric conditions. The name tricyclics comes from their three-ring molecular structure. Although TCAs have mostly been replaced by SSRIs due to their fewer side effects, TCAs are still used for severe depression. So, how do TCAs work? Well, it's a bit complex, but the main thing they do is block the reuptake of norepinephrine and serotonin in the presynaptic terminals. This means more of these neurotransmitters stay in the brain, which helps improve mood and reduce depression symptoms. In the long term, TCAs can alter the binding of NMDA receptors, which might provide analgesic effects by inhibiting neuronal hyperexcitability. Additionally, TCAs block several types of receptors, including cholinergic, adrenergic, and histaminergic receptors, as well as ion channels like sodium channels. This blocking action is the reason behind their various side effects. For instance, blocking histamine H1 receptors can cause drowsiness. Blocking muscarinic receptors can lead to dry mouth and blurred vision. Blocking adrenergic receptors can cause low blood pressure. Some TCAs also have a quinidine-like effect, which can suppress heart function, so careful dosage is crucial. Let's do a quick quiz. Do you know how TCAs work? Correct! TCAs mainly work by inhibiting the reuptake of both serotonin and norepinephrine. They also block cholinergic, adrenergic, and histaminergic receptors. This blocking action is why they have various side effects.
Lastly, about monoamine oxidase inhibitors, as we saw at the beginning of this video, they were initially used to treat tuberculosis. Monoamine oxidase inhibitors, or MAOIs, work by blocking the effects of an enzyme called monoamine oxidase. But first, do you know what monoamine oxidase actually is? Monoamine oxidase is an enzyme that breaks down important chemicals in the brain called neurotransmitters, such as norepinephrine, epinephrine, serotonin, and dopamine. There are two types of this enzyme, monoamine oxidase A and monoamine oxidase B. MAOA mainly breaks down serotonin, norepinephrine, and epinephrine, as well as certain substances in food like tyramine. MAOB mainly breaks down dopamine and phenethylamine. This is a good opportunity to review the metabolic pathway of monoamines. Monoamine oxidase inhibitors block this enzyme, leading to an increase in neurotransmitters in the brain. This helps improve mood and other symptoms.
You might have heard the question, what foods should you avoid when taking MAOIs? The answer is, foods containing tyramine should be avoided by patients taking MAOIs. But why? Because normally, tyramine is broken down by monoamine oxidase in the gut and liver. But when someone is taking MAOIs, tyramine is not broken down and its levels in the body can become too high. Tyramine is thought to indirectly trigger the release of norepinephrine. When this happens, it can cause blood vessels to constrict, leading to migraines or high blood pressure. Tyramine is found in foods that are aged or fermented. This includes items like pepperoni, hot dogs, wine, and condiments like soy sauce. It's important to be mindful of these foods, especially if patients are taking monoamine oxidase inhibitors. Let's do a quick quiz. Do you know how MAOIs work? Correct! MAOIs inhibit the breakdown of neurotransmitters. Tyramine is thought to indirectly trigger the release of norepinephrine, so it's important to be mindful of tyramine-rich foods while taking MAOIs. Thanks to the tireless efforts of researchers, antidepressants are continuously evolving. But there are still questions about whether the chemical imbalance hypothesis is truly valid. This is because, even though antidepressants increase monoamine levels in the synaptic cleft, it takes several weeks or more for symptoms to improve. One theory suggests that the rapid increase in monoamine levels might reduce receptor activity. Additionally, the activity of presynaptic neurons may increase due to the inhibition caused by antidepressants. It might take several weeks for this state to stabilize. We should always respect the researchers who have discovered what we believe now. At the same time, we should always be prepared to accept changes flexibly, based on new discoveries, in the next 5 or 10 years.
Subscribe for more insights on how medications work.